Fascicles are composed of myofibers, which is this. Myofiber is the same thing as a muscle cell. Muscle cells are multinucleated. That is the nucleus right there. Muscle cells also contain mitochondria, which is right there. The muscle cell is wrapped in a layer called the sarcolemma. This is a myofibril. The sarcolemma has invaginations which create T-tubules, which are this blue line right here. The T-tubule and the terminal cisternae create the triad. The terminal cisternae contain most of the calcium of the sarcoplasmic reticulum. This is a sarcoplasmic reticulum. This is where all the calcium is stored, and right here. The molecule that holds the calcium in the sarcoplasmic reticulum is calquestrin. So this is filled with calquestrin. Inside the myofibril are myofilaments. These.
So the filaments create certain bands that can be seen. You have the H band, which is going to be just the thick filament here. And then you have the A band, which is created of both thick and thin filament. And then you have the I band, which is over here, which is just going to be thin filament. So between each I band is a Z band of darkness, and it's between the two Z bands that is considered a sarcomere. Okay. So this is the structure from which our nerve impulses are going to go. First you have the axon, and then it goes into the axon terminal. And then right here is the synaptic cleft. And then we have our motor end plate, which goes into the T-tubule, where the action potential is propagated within the muscle cell. And then we have the terminal cisterine. And in here you can see that the purple is the calcium already stored, along with the calquestrin keeping it in. And right here within the muscle cell we have the potassium, which is our green. And then in synaptic cleft we have more calcium. And then we have sodium. And then the red is going to be our acetylcholine esterase. And then inside the axon terminal, we have our acetylcholine. So the action potential comes down into the axon, and it's going to stimulate calcium channels, which is going to allow the calcium to come into the axon. This is going to allow the synaptic vesicles filled with the yellow acetylcholine to come out and fuse with the membrane and then go out into the synaptic cleft. Once it diffuses into the synaptic cleft, it is going to attach to receptors on the motor end plate. Okay. Once they refuse the receptors, they're going to activate sodium-potassium pumps that are going to allow potassium to leave the motor end plate and allow sodium to come inside. So although potassium is positive and sodium, more positive is coming in with the sodium, so the membrane is depolarized, which generates an action potential. However, while this is going on, the acetylcholine is starting to get broken down by the acetylcholinease, which allows the components to be brought back into the axon. If it wasn't for the acetylcholine esterase, then acetylcholine would be constantly keeping the channels open, which would cause tetanitis in the body. So now that the action potential has been generated, it goes along down the T-tubule. So it allows for the release of calcium from the sarcoplasmic reticulum, and which is let go by the calquestrin. At this point, the calcium that's been released is going to bind into troponin, which allows for the actual contraction of the muscle. So here we have it zoomed in even more with the actin and the troponin complex on it, which is blocking the binding site from myosin. So what happens is calcium comes in and it moves the troponin myosin complex and allows for the bonding sites to be free. Here we have one of the individual fibers. We have axin, and it's wrapped around with the tropomyosin complex with individual troponin on it. So here we have the myosin, which is in its high energy state with the heads cocked that way. And once again, we have our actin that's wrapped around with the tropomyosin complex with the troponin. So what happens is we're going to have calcium move in and attach to the troponin on the tropomyosin complex. And what it's going to do is allow it to be shifted, which allows that the myosin heads to bond to the active sites. As you can see, the actin has been shifted over with the contraction. You can see that as compared to the myosin that's still in the high energy state. So now that it's been contracted, what needs to happen is for the calcium to be removed so that the myosin can go back into, can be released from the actin.